Okay, good evening, student. Today we will start about the topic protoplasm. First, I would like to start that what is the meaning of term protoplasm? Protoplasm is basically a kind of living liquid. What I'm calling it living liquid. Living liquid means a liquid they have a capability to sustain living bodies. So first, I would like to clear the definition of protoplasm. Protoplasm is the physical basis of life. It is just what it is, the physical basis of a famous scientist Perkinjay give the term give the term protoplasm. So first I would like to clear that what is the protoplasm is the physical basis of life, and we can't imagine the life without the protoplasm. Later on, protoplasm can be understood according to the working scenario and according to its composition. First, I would like to discuss about the composition of the protoplasm. Composition of the protoplasm. It is mainly composed of four atoms, mainly composed of nitrogen, oxygen, carbon, and hydrogen. These nitrogen, oxygen, carbon, hydrogen will form near about 92 will form near about 90 to 92% part of the protoplasm. 92% part of the protoplasm. And what about the rest part of the protoplasm? The rest of the protoplasm is formed by the rest of the protoplasm. The rest of the protoplasm is formed by minerals and ions. Minerals and ions. Later on, we'll discuss about what is the protoplasm aggregation. Protoplasm is mainly composed of organic and inorganic substance. Among this inorganic and organic substance, if I discuss about all of the body protoplasm, the body liquid will be consist of mostly the body liquid will consist of mostly will consist of mostly water. There are near about there are near about 70 to 75% water present in a body, and rest 30 to 35% contain organic substance. Rest part will be organic and minerals will be organic and minerals. Among rest 30 among rest 30%, there is 14% part will be formed by protein. So first we have to discuss about the term protein, because protein consists of near about 14% of the protoplasm part, and it is the maximum percentage of organic substance present in a protoplasm. Percent of organic substance, maximum percent of organic substance. Organic. First, we'll discuss about the protein. Protein is basically a kind of building block of the body. These are also termed as a building block of the body. Block of the body. These are known as what? This is known as building block of the body. This is important because our body functions is mainly controlled by enzymes, genes, DNA. These all enzymes, genes and DNA, these are formed by the proteins. So first we'll discuss about the structural and functional part of the protein. So first, what is the unit of the protein? If I would like to start about the unit of the protein, the unit of the protein will be, the unit of the protein is amino acid. This is what? This is amino acid. So I can simply say that, so how the protein will form? Protein will form by the arranging, or we can say the aggregation aggregation of the amino acid chain. Amino acid chain is protein. It was discovered by a scientist Berzelius. It was discovered by the famous scientist Berzelius. So first we have to study about the structure of amino acid. So first I would like to study about the structure of amino acids, and later on we study about the bonding of amino acids. First, the structure of amino acid. As suggested earlier, it's an organic substance contain carbon. It's an organic substance which contain carbon, which contain the carbon molecule. Next is what if I discuss about the structure of a carbon molecule. Carbon is I would like to start with this. This is the carbon molecule, and this is the fourth bonding of that particular carbon. This is carbon number 1, carbon number 2, 3, and 4. We all know that carbon show 4 tetravalent valency of a covalent bonding. And among these 4, the first is substituted by hydrogen, second is substituted by R is termed as alkyl group. What's that? It's alkyl group. This is alkyl group. And another side is formed by this is known as and H2, and the fourth one, that is the C double O L H. This is the carboxylic acid group. Carboxylic acid group. This is what? This is amine group, and this is hydrogen. This is what? This is hydrogen. So this is the basic structure of an amino acid. If I discuss about this amino acid structure, this is the structure of amino acid. And what is the importance of these amino acids? I would like to study about this amino acid structure. This is all about the structure of amino acid that I form in a box around these all structure. This is the structure of amino acid. Amino acid is basically work like a deuterium. Amino acid is basically what? Amino acid is basically a deuterium in which acid and base in which acid and base both are are present. Then I'll discuss about how this amino acid will form the deuterium. This is formed because of that, like that. Suppose I draw again a simple molecular structure of that. We all know that this is the what? O, this is H, this is N. We all know that there is a lone pair of electron. This is hydrogen and this is the hydrogen. We all know that the electronegativity of oxygen is quite far than hydrogen. The electrons of the bond will be attracted by oxygen. Then this oxygen, after getting the electrons, will become a negative charge, partial negative charge. And this hydrogen will remove from here, will remove. And this removing hydrogen will be accepted by this nitrogen group because of the extra lone pair which I am mentioning here, which is the extra lone pair here. So the structure of the deuterium can be written like that. Suppose I write the structure of a deuterium here. The structure of deuterium can be written like that. H, this is R, this is C, O, O negative, and H, 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 and right now it will become partial positive charge. So this is the structure of Jeter ion. So here we can simply say that this Jeter ion will exactly a very important part of the protein structure, because it's a structure. We can simply say that this is the this is showing what this is showing the grouping of acid and base both. Next we'll discuss about that what next we'll discuss about that how this Jeter ion will form the protein molecules. Later we'll discuss about the formation of protein molecule. There is a formation of protein molecule. How this molecule of protein will form? Formation of protein. Here we have one amino acid group C H R O O H and H H plus again a consecutive amino acid will be here H C H. Suppose I'll write here R1, this is R2, next C double O H. So when the molecule of protein will form, there is a removal of water molecule from this side. And this water molecule, I can write here this water molecule minus of H2O from this reaction. There is again a formation of bond. This bond is basically, first I write the structure of a remaining part, which is the current remain here. This is what C, H, this is R, this is and this is H, this H amine group, C double bond O, single bond, and H, C, H, R2, R1, O, H. So here, this bond, which I am denoting here with that particular structure, that bond that is known as what? This is the bond which connect two molecules of a amino acid, and this molecule is this bonding is termed as what? Peptide bond. Right? So we can simply say that this peptide bond is very important because peptide bond can helpful in bonding the two amino acids. Today we have discussed about the amino acid groups present between the two between molecules of proteins, and there is a bond between two amino acids. Recently termed as what? This is termed as peptide. So later we'll discuss about the next carbohydrate protein, carbohydrate fats, etc. Okay, thanks a lot.